Okay, hello people. So this is the last day that we're going to do nitrogen catabolism for proteins. When we come back from the exam, we'll talk a little bit about metabolism regarding nucleotides, since those also have nitrogens in them. But today's is a little bit different than what we've been talking about. Today is specifically going to be degradation of non-dietary proteins. So why would we want to degrade a non-dietary protein? First of all, what are some examples of non-dietary proteins? These would be things like hemoglobin, myoglobin, maybe pepsin. These are the proteins that are made by your body via the process of translation, and oftentimes these proteins get damaged and then they need to be degraded. Okay, so that's really what this one is going to be about, is about pathways for degradation of non-dietary proteins. And proteins have half-lives that can vary widely. Can vary from anything from 30 seconds all the way up to days. And again, if it's a damaged protein, it's going to be targeted for degradation very, 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 very quickly. But there's a few things that determine the protein's half-life. One of them is the N-terminal sequence identity. One of them is a PES sequence, and then the third one is where we're going to be spending most of our time, which is on the pathway of degradation if the protein has been tagged with this other protein called ubiquitin. But first, let's just briefly recall N-terminal sequence, N-terminal amino acid sequence identity and PES sequences. We did run into those in Biochem 1 just a little bit, so I'm going to bring them back up. So it turns out that the identity of the amino acid, I'm just going to read this, that follows the N-terminal methionine seems to be important in determining the half-life of a protein for bacteria and eukaryotes. Okay, so what does this mean? Go away. So when a protein is being translated, the beginner or the very first amino acid that gets put on is this methionine. That's why we call that N-terminal methionine or N-met. And then anything after that, let's say it's arginine, and then proline, and then lysine, and so on and so forth. This would be residue number 1. This is residue number 2, 3, 4, and so on. It turns out that there's like this hierarchy in the cell for primary sequences where this methionine gets cut off. And then there's a brand new N-terminal sequence identity. So if this gets cut off, let's see, so that methionine, he gets cut off. And again, there's a hierarchy in the cell. This will be a post-translational modification. That makes this arginine the new residue number 1, or the new N-terminal sequence identity. Okay, it's the new N-terminal residue. If it happens to be an arginine, oh look, I didn't even do that on purpose. That would make this primary sequence targeted for degradation or its half-life very, very, very short. It will only last in the cell for approximately 2 minutes, which isn't very long. You can probably imagine that hemoglobin and myoglobin are somewhere up here because those proteins last for a very, very long time. Okay, that's N-terminal sequence identity. Another thing that seems to be important are these PES sequences. So proteins that contain sequences that are rich in proline. And remember, proline's one-letter code is P. Glutamic acid or glutamate, one-letter code is E. Serine, S. And threonine, T. And if you notice that these spell pest when together. So proteins that contain sequences that are rich in proline, glutamic acid, serine, or threonine tend to have half-lives that are less than 2 hours. Okay, so those are two things that kind of determine a protein's half-life. Now, when a damaged protein needs to be degraded, or any of these other proteins need to be degraded, they're going to be degraded through this ubiquitin pathway. And we're going to talk about that next. So there's this protein, and he's just kind of a little guy. He only has 76 amino acids. And his name is ubiquitin, and it's not a he. Ubiquitin, which is often abbreviated capital U, lowercase b. This little teeny tiny protein is ubiquitous, which is why it's called ubiquitin, which means that it's present in all cells in eukaryotes. And it's particularly a well-conserved protein, which means evolutionarily not much has changed with this protein. This figure right here, which is a ribbon diagram of ubiquitin, it's mostly beta sheet. It's got a little tiny alpha helix there. I can't tell whether that's a 310 helix or an alpha helix. It looks a little bit wider. And you'll notice that there are some lysines that are sticking out here, and those are going to be important, so pay attention to the lysine. So it's this little teeny tiny guy. It's called ubiquitin because it's ubiquitous, or present in many, well, actually it's present in all the cells, because all cells either produce damaged proteins or have proteins that their function is no longer needed, so they need to undergo degradation. And this degradation, again, is not dietary degradation. These aren't going to be turned into urea and excreted. Instead, they're just going to be turned back into amino acids that are going to be recycled for translation again. All right, so here's the process. There are 3 main proteins that are needed. I'm just going to zoom in over here. So we need, actually, maybe I should just start with this. We need ubiquitin, UB. Is the abbreviation for ubiquitin, and again, it's a very small protein, so when you see it, it will be the tiniest one there. So this is ubiquitin. Okay, he's just simply a tagging protein. Okay, so when we say a protein has been tagged for degradation, the tag, or the little flag that says, hey, I need to be degraded, the little flag or the tag is ubiquitin. So anything that's been, we would call it polyubiquinated, which means as many ubiquitins has been tagged for degradation. It's basically a signal that says, hey, time to be recycled. Okay, and then we have E1, and E1 is the ubiquitin activating enzyme. So I'm just going to say UB for ubiquitin activating enzyme. E2 is the ubiquitin conjugating enzyme. And then E3 is the ubiquitin ligase. Oops. Okay. And then we have the protein. So these are the 3 things that you need in order to put a ubiquitin on the protein that is getting degraded. And then we actually need the protein that will be degraded. And for whatever reason, the ubiquitin people, they call this the substrate. Okay, so the protein that's going to be tagged or get a whole bunch of ubiquitins on it for degradation to be broken up into all of its little amino acid pieces will be called the substrate. So there's a process diagram here that I've got, and we're just going to go through each one. There's really only 3 steps here. Well, yeah, 3-ish steps. And then the last one is just lather, rinse, repeat, go around and around and around again. All right, so step number 1, is to put the ubiquitin on E1. So the ubiquitin, oops, I don't want that color. Oh, blue. Or green. No, blue. I want blue. So the C terminal, the C terminus, which is this carboxylic acid, and you can see it right here, the C terminus of ubiquitin is linked to a cysteine residue, which has an SH on the side chain, is linked to a cysteine residue. So there's a cysteine right there. I'd like you to, when you're taking notes or when you're practicing this, I'd like you to propose a mechanism for this linkage. For the linkage, for the connection between the carboxylic acid and the thiol group. So now we have what looks kind of like an amide functional group, but this should be, I don't know if you can tell, but that should be a double. That's a carbonyl carbon to an S. So it looks kind of like an amide, and instead of a nitrogen, you have a sulfur there. Okay. So again, E1 is the activating enzyme, so it's just going to link the ubiquitin to E1 covalently. And then the second step is to basically swap places. So the E2 will link to ubiquitin, also using a cysteine residue. And then E1 is released. Okay. So we've just basically switched partners, this thiol group. Again, you can probably propose a mechanism here, comes in and attacks. And again, this should be, I'm sorry for how pixelated this is. This is the best drawing I could find though. So now E2 is linked to ubiquitin. E2 then connects to E3. So I'll say E2/UB or -UB, indicating that those two pieces are connected, binds to E3, which is bound to the substrate protein. Again, I don't want you guys to get too confused here. The substrate, this is the protein. I'll call it the damaged protein, call it whatever. This is a protein that needs to be recycled. Okay. And then in the, almost to the last step, the ubiquitin is transferred to the substrate via a lysine residue on the substrate. So this residue right here on the substrate, again, is going to come and grab that ubiquitin, breaking the bond between the carbonyl carbon and the sulfur group. And what you get as a result is a new amide bond, but you're linked to the side chain of a lysine. Okay. And then in order for this substrate or this protein to be completely degraded, they actually have to put many ubiquitins on. So you're going to repeat the cycle to make a polyubiquitin chain. Okay. And what I'm going to say about this is that the linkage of ubiquitin to ubiquitin is the same. It's just also there's lysine residues coming off of the ubiquitin. So in the second round, the E3 would be attached, the substrate would be a ubiquitin, and would just keep like, well, the substrate wouldn't be ubiquitin, the substrate would be this, but the ubiquitin would still move from here down to the substrate, but it would already have the ubiquitin on it, but it's still going to a lysine. Woof, that did not make sense. Let me see if I can move forward to the next piece and see if I can make that make a little bit more sense. So, yeah, move forward. Okay, so the first ubiquitin is attached to the substrate. Okay, then this piece right here will go back around again, and then the second ubiquitin comes in and is linked C-terminus to a lysine on the first ubiquitin. So what I'm really trying to say is that every time a new ubiquitin is added onto the tagged substrate, it's always moving to a lysine residue, and you're always making a new peptide bond. So you get a polyubiquinated protein, and it just keeps going around and around again. So those lysine residues on the ubiquitin are highly conserved. Okay, so now we have a polyubiquinated substrate, or a tagged protein for degradation. Where it will go next is this ginormous protein that's called the proteasome. Okay, this is a humongous protein complex, and this is one of the most simple diagrams that I could find of it. Every bubble here is a protein subunit. So there's for sure quaternary structure, and there's many subunits, and we're going to go through all the different pieces here because it's actually a really interesting protein. It's kind of like, I always think of it as like a garbage can. Okay, so here's like the can piece, and here's the lid. Okay, and the inside of this is actually kind of hollow, but interestingly, the bottom part here is also a lid. Okay, so basically you're going to have a protein going in to the can, the lid will close, it will get degraded, and then the lid on the bottom will open to let the protein that's been degraded, it will let it out. But let's go over the pieces. I'm just going to get rid of this so it's not so messy. Okay, so this is the proteasome is in all the pastel colors. And then the rest of this is a substrate that's been added to this. So here is the protein substrate. This is the protein that has been tagged for degradation. So it's going to go from its tertiary, quaternary structure all the way into single amino acid pieces. These little red spheres here are the ubiquitins. Okay, so the ubiquitin attached to the substrate is what allows, whoa, what happened? This allows for attachment. Okay, and where it's attaching is what's called the 19S regulatory particle. Regulatory particle. And again, there are two of these. This is also a 19S regulatory particle up top. The middle piece here in the blue and the gray is called the 20S particle. And the function of that, I didn't quite leave myself enough space. So the function of the 20S is the proteolytic cleavage. Okay, so here inside of this, which is, again, it's humongous and it's like a hollow tube on the inside, it has active sites that are like chymotrypsin. And now I'm going to put chymotrypsin-like because it's not chymotrypsin, but it acts like a chymotrypsin. Trypsin-like. And this third proteolytic function, where did I put it? I didn't want to misspell this. Alright, so it's called peptidyl glutamyl peptide hydrolysis, I think is what the last part is. This is often abbreviated PGPH. So basically the core of this, the 20S core piece, is the part where it's going to be broken into all of its little amino acid and little peptide pieces. And then the entirety put together is called the 26S, or the complete proteasome. Okay, so those are the parts. This is an image that I really liked a lot because I feel like it shows some interesting functions here. So it's got the 19S piece labeled as the lid, and then the core here is the 20S, and then also just remember there's another 19S down at the bottom here that they aren't showing. But what's really interesting about this, so it shows the substrate that has the polyubiquitin tag. The substrate binds, and again, the substrate is a protein. It binds because it has the polyubiquitin. The ubiquitin is able to leave. And then this yellow piece, also in the 19S, helps to unfold the protein. And it's an ATPase, so what that should be telling you is that this requires ATP in order to function. And then as the substrate or the protein is unfolded, it's able to enter into the proteolytic site where it's broken into all of its little pieces, and then these pieces will leave, and the pieces are single amino acids and peptides. And they will go on back to translation to make new protein. So again, not going to go to the urea cycle unless for whatever reason there's a crazy amount of protein around. But they're going to be recycled so that they can go back into, so maybe I'll say this, so the amino acids or peptides that exit the proteasome will be recycled. How do I say this? So it will be recycled not during translation, but recycled for translation of new proteins. Okay, there's a YouTube video link here that I'm going to link into your into the course website so that you can kind of see the process in motion. You can see the lid opening, the lid closing, and the process of getting this polyubiquitination. But that's basically it. Those are all the pieces that I'm going to focus on. So again, proteins, these are non-dietary proteins. This would be like chymotrypsin or trypsin or lactase. These are damaged proteins, or proteins that you no longer need their function. Maybe it's hexokinase, maybe it's lactate dehydrogenase. They'll be tagged for degradation by ubiquitin. And you'll need many ubiquitins to tag a substrate. Again, the substrate is the protein that's getting degraded. So if you want to think of this as hexokinase in green here, we don't need hexokinase anymore. It'd be tagged for ubiquitination. The ubiquitin allows this hexokinase bubble here. Again, I'm just making up a protein. The hexokinase to attach to the proteasome. It's unfolded using ATP. That unfolded chain enters into the proteolytic site of the proteasome, where it's degraded into its individual amino acid pieces or its peptides. And then the bottom part here, which is also a lid, can open so that those peptides and amino acids can be released back into the cytosol so that the ribosome can use them again in translation. And that's it. I'll see you later. Bye.